Okay, so there is so much buzz right now around psilocybin, the active compound in magic mushrooms, and its potential for mental health care. It really feels like a revolution is underway. But with everyone so laser focused on this one chemical, it makes you wonder, what about the mushroom itself? The whole organism? Well, a recent scientific study decided to dig into that very question. And honestly, what they turned up is pretty astonishing.
So just listen to this. This is the big bombshell conclusion right from the paper. They use words like inaccurate, irreproducible, and inconsistent. In the world of science, those are really, really strong words. It kind of suggests that a lot of what we thought we knew might be built on some pretty shaky ground. So what we're going to do is play scientific detective for a few minutes and unpack exactly how they got here. Our investigation starts with what you could call a massive identity crisis.
All right, part one, the identity crisis. I mean, think about it. Before you can even begin to analyze a mushroom's chemistry, you have to be 100% sure what kind of mushroom you're actually holding. It's step one, the absolute basic foundation. And right here is where the researchers found their first huge problem.
This really is the heart of the matter. See, scientists rely on these things called fungaria. They're basically libraries of preserved fungi for a lot of their research. And they have to trust the labels on the jars. But should they? Well, this study put that trust to the test, using modern DNA barcoding.
And here's the kicker. What they found was that roughly 13%, yeah, that's more than 1 in every 10, of the specimens labeled as being in the Psilocybe genus weren't Psilocybe at all. The DNA told a completely different story.
And this isn't just some minor little mix-up for mushroom nerds. Look at this. The label on the jar said Psilocybe, but the DNA results came back saying, nope, this is actually something like a Diconica or a Gallerina. And here's why that's such a big deal. Those other kinds of mushrooms aren't even known to make psilocybin. So just imagine all the past chemical studies that might have been done on these mislabeled samples. They would have been totally misleading from the get-go.
And it gets worse. The problem isn't just with these old jars in museum collections. The researchers found that the public DNA databases, the very tools that scientists use to identify new samples, are also full of mistakes and mislabeled genetic sequences. It creates this vicious cycle of confusion that makes it incredibly hard for anyone to get a solid, reliable ID. But okay, even if you do get the right mushroom, you run into a second problem. And that brings us to our next case file, the fading evidence.
So part two, the fading evidence. Let's say you hit the jackpot. You've got a correctly identified psilocybe mushroom from a collection. The next big question is, after sitting on a shelf for, 30, 40, 50 years, does it still have the psychoactive compounds we're looking for? Or does the magic, so to speak, just fade away?
To figure this out, the scientists analyzed a whole bunch of specimens that had been collected and stored over a nearly 50-year period. I mean, we're talking about samples from the 1970s all the way up to today. They were specifically looking for psilocybin and psilocin to see how well these molecules hold up over time.
And what they found was, well, it's complicated. The results were all over the place. Turns out, stability really depends on the exact species. On the one hand, you have something like Psilocybe cubensis. That one turned out to be surprisingly tough. Even after almost half a century, those specimens still had a lot of psilocybin. But then you look at Psilocybe cubensis, which is one of the most common and widely studied species out there, and it's a different story. For those, the compounds just fell apart.
This is such a great example of what they found. They tested a 42-year-old sample of P. cubensis that luckily had been chemically analyzed way back when it was fresh. Today, it has less than half the psilocybin it started with, and the psilocin, which is what psilocybin turns into in your body, was totally gone, vanished. It just proves that the chemical profile of these preserved mushrooms can be a totally unpredictable moving target. Okay, this all leads us to our third and final mystery. So far, we've been talking about the things we thought we knew, but what about all the stuff we have no idea about? The research team decided to look beyond just psilocybin and explore the mushroom's entire chemical world.
The way they did this was by studying something called a metabolome. Just think of the metabolome as the complete chemical fingerprint of any living thing. It's not just the one or two famous compounds, it is every single little molecule that organism makes.
So when they did this for just one species, Psilocybe cubensis, they detected 1,465 unique chemical compounds. Let that sink in. Most of the research you read about focuses on maybe 2 or 3 of those. So you have to ask, what on earth are all those other chemicals?
And this is where things get truly wild. Of all those compounds, nearly 1,500 of them,
Guess how many they could actually identify and match to known chemicals in existing databases? About 0.2%. That means a staggering 99.8% of the mushroom's chemical fingerprint is what they call chemical dark matter. We have almost no idea what these molecules are or what they do.
But hidden inside that dark matter, they found some really fascinating clues. They found signals for compounds that look a lot like serotonin, which is a key neurotransmitter in our brains. They even found something that looked a bit like trazodone, a synthetic antidepressant. Oh, and they also found a chemical that the mushroom probably just soaked up from the coconut stuff it was grown on. This tells us two huge things. One, there are probably other biologically active compounds in there we know nothing about. And two, these mushrooms are like sponges, absorbing things from their environment, which just adds another layer of chemical complexity to the whole thing.
So, we've got an identity crisis, we've got fading evidence, and we've got a vast, hidden universe of unknown chemicals. So what? What does this all actually mean in the real world? Why does any of this matter for the future of psychedelic therapy?
Let's just break it down into 3 key points. First, we're talking about faulty foundations. All this misidentification means a huge chunk of past research might just be plain wrong. Second, this creates all kinds of regulatory riddles. How are you supposed to write clear, evidence-based laws for these things if their chemical makeup changes so much over time? And finally, this points to some massive therapeutic unknowns. When a person consumes a whole mushroom, they're not just taking psilocybin. They're taking a cocktail of nearly 1,500 different compounds, and we have almost no clue what the rest of that stuff does.
And that brings us right to this crucial quote from the paper. The scientists bring up the very real possibility of an entourage effect. That's the idea that all these other unknown compounds might be working together, synergistically, creating an effect that's totally different from what you'd get from pure synthetic psilocybin. The whole mushroom might truly be greater than the sum of its parts.
So I think the big takeaway is this. As we stand here on the edge of a new era of psychedelic medicine, this research is a super important reality check. It's challenging us to look past the one famous molecule and to start doing the really hard work of understanding the entire, deeply complex, and still very mysterious organism that produces it.